thing right here.
AV node.
This is the AV node. Number five was identify these fibers right here. Purkinje fibers. Number 6 asks the function of this structure. What is it pointing to?
SA node.
It's pointing to the SA node. What does that do?
Activates.
It initiates or starts the impulse or heartbeat or whatever. Number 7 is if a person has A positive, who can they donate to?
A pause.
A positive and
AB positive.
AB positive. If a person has AB negative, excuse me, who can they receive from?
A negative and O.
They can
O
receive from
negative.
all negatives. A negative, B negative, AB negative, and O negative. Remember that this is the universal recipient, so that they can receive from like all blood types. But they're negative, so they can only receive from negative. They can't receive from positive. Okay. Number 9 is asking to identify this little white thing. Yeah. Identify this little white thing in here.
The simulator, aortic simulator. Valve.
It is a semilunar
Semilunar
valve.
pulmonary.
What is this vessel?
Pulmonary
This is the
trunk.
pulmonary semilunar valve. Okay. And number 10, it's pointing to the inferior vena cava, but the question was, where does blood come before this?
Systemic.
From the systemic circulation, or you could have said body, either one of those. Okay. Any questions on this? Okie dokie. Hold on, let me go grab something.
So when I thought when it said 4 years, it's like when it's all done, what comes.
I thought it was like what comes next.
So I talked about the constriction of the atrial because I'm thinking it's the next thing is B wave.
I think I forgot.
Right.
So,
Yeah. I probably made like a 70, 65.
So in just like we're gonna go through the different types of vessels first, but after we go through the different types of vessels, we're gonna actually draw the vessels and like kind of do road maps. So I know that a lot of you guys like just like have your lab assignment, but if you need extra paper to like draw what we're doing today, we're gonna do like all the drawings all day today. So if you need paper, you can come up here and grab some while I get situated.
Thank you. Thank you.
And there's more paper in the back, so if we run out, we'll just get it
Section 21.
I don't know. I'm not sure where we're at.
We'll find out.
Okay, so today we are going through the blood vessels. We're going to start on page 93. Page 93. And let's, basically page 93 is everything that we did in lecture today. So I'm sorry for the double lecture today, but it is what it is. So, first things first, is the different layers of the heart. Remember that we said that there, or not the heart, layers of your blood vessels. We said that the innermost was called the tunica intima, the middle layer is the tunica media, and the outermost layer is the tunica externa. So again, don't forget that the difference between arteries and veins is the lumen size when we start from the inside. your tunica intima of an artery is going to be relatively circular, and it's going to maintain that shape. And you can see how small it is relatively compared to if we had a vein. The lumen would be very large, and it would be kind of collapsed, okay? So you can see that it's in a perfect circle over here, it's kind of collapsed in a weird shape over here, okay? Now, this is made both out of simple squamous epithelium. And this is kind of like when we get to the histology, not all the time, but sometimes when you have the vessels, instead of it making like a perfect circle like this, you might see it kind of have like this coiled network or a coiled shape to it. It is still simple squamous epithelium, but it's just under high tension and high pressure that it like collapses and recoils, okay? So that's the tunica intima. The tunica media is made mostly out of smooth muscle. Now, if you remember, we said that the arteries are under high pressure situations, so the tunica media is going to be very thick. You're going to see this huge outer layer, or middle layer, of muscle. Very thick middle layer. When we look at the tunica media of a vein, it's going to be much thinner. So you are going to have it, but it's not as significant as your artery over there. Does that make sense? Okay, you have the same layers, it's just thicker in an artery versus a vein. The last layer is your tunica externa. It's made out of dense irregular connective tissue, and it's very diffuse, meaning that it, like here, you can see that the layers are very like condensed and put together. The loose connective tissue is just going to be kind of spread out everywhere. So you're just going to kind of see like a random outer surface out here. And it's not going to have any set shape to it. It's just kind of like the surrounding structure out here. And it's going to be the same thing in the vein. You're going to have this random like outer structure here. There's no set shape to it, but it's an outer layer again to anchor and hold this vessel in place. Okay? Any questions on the differences between arteries versus veins? All right, so let's move down to the next table. Still on page 93. We're going to talk about the different vessels. So let's do a kind of like a flow chart to show the different vessels, starting with the heart up here. What did we say in lecture was the first type of vessel that comes out of the heart? The aorta, but what kind of vessel is that?
Elastic.
It's an elastic artery, okay? So coming out of the heart, first you're going to have elastic arteries. Then as we move away from the heart, you're going to have muscular arteries. Then as we get even further, and as we get closer to the capillaries, we're gonna have arterioles, which leads to the smallest vessel or gas exchange called capillaries. Okay? What was the opposite structure of arterioles? Venules. So the capillaries are going to get bigger and they're going to form venules. And your lab manual says that there's like small to medium arteries and then small to medium veins and then large veins. You don't have to tell the difference between the different size veins. They're all veins, okay? So from venules, it goes to veins, and then it goes back to the heart, okay? So your elastic arteries, as we come out of the heart, again, are under the highest pressure, so they're gonna be mostly made out of elastic connective tissue, okay? The muscular arteries, like the name implies, is gonna be mostly made out of smooth muscle. So the name literally tells you what tissue it's mostly made out of, okay? Arterioles are gonna be made of simple squamous epithelium, but we're not gonna have the 3 full layers. We're typically just gonna have, like I said, that simple squamous epithelium and maybe a smooth muscle cell or 2 surrounding that, okay? By the time we get to the capillaries, this is gonna be composed only of simple squamous epithelium. When we get up to the venules, we're gonna start to have, again, the 3 layers. We have tunica, intima, media, and externa, but they're just a little bit larger. And then the veins are gonna be even larger venules. So again, they're gonna have the 3 major layers, okay? So if we look at this, the function of all of these layers, or the function of all of these vessels, is to propel the blood to the next type of vessel. So the function of elastic arteries is to send blood to muscular. The function of muscular is to send blood to arterial. Function of arterial, send blood to capillaries. Capillaries send blood to venules. Venules send blood to veins. So if you know this circuit right here, you know the functions of all of these vessels, okay? Now, remember we said that there were different types of capillaries, right? There's the continuous, the fenestrated, and the sinusoidal. So the difference between these again are the gaps or the spaces in between them and what they allow through. So the continuous, just like the name implies, you're gonna have the cells literally touching each other, and only the smallest substances, like your gases, can go through, okay? So it's permeable only to small things like oxygen and carbon dioxide passing through here, okay? Your fenestrated, like we said, are going to be a little bit spaced out. So instead of the cells touching each other like that, you're gonna see a little bit of a gap in between here. So now, not just gases, but small particles like glucose right here. See how glucose is small enough? It can fit in between these bigger gaps in the in the capillary. Your sinusoids are going to be the largest gaps. You're going to see a cell here, you're going to see a cell here, and another here. These gaps are so big that proteins and red blood cells can fit through these gaps right here, okay? So it's just a matter of what do you want to be able to pass through? Do you want cells to pass through? Then you're gonna have the sinusoid. Do you want only gases to pass through? Then you're gonna have continuous, okay? Now, I just wanna point out that continuous is the most common type. Like this is what we have pretty much most places in the body. These 2 are only in like special places like the kidney and other places, and the spleen and stuff, okay? Any questions on capillaries? Now, what did we say the word anastomosis meant? It's a connection or a junction between 2 vessels. So the word arterial anastomosis means it's a connection between 2 arteries. So what you would see is that instead of going from like this, like down to the capillaries, it would go out to these other arteries over here before going down here. So it's basically like adding an extra step. We're adding more arteries to like the circulation. If you have a venous anastomosis, then the vein right here would go to another vein before going back into the circulation. Does that make sense? Okay, so arterial is artery to artery, Venous is vein to vein. What do you think would happen if you have an arteriovenous anastomosis?
You have both.
Between the artery and vein. So it basically, instead of going to this capillary bed right here, it goes directly in this direction right here. Okay? What did we say was special about a portal system?
It has 2 capillaries.
There's 2 capillary beds. So what you would see is that instead of it going from arterioles, capillaries, venules, you would have another capillary bed right here. Capillary. And the blood would go to this capillary bed, and then it would go to the venules. Okay, so it goes from arterioles, capillary 1, capillary 2, then back to the normal circulation. Does that make sense? Any questions on these types of vessels or the different types of pathways? Okay, so that's pretty much it on page 93. If you turn to page 94, I know that that list looks scary. These are the vessels that you guys are responsible for. Remember how in AMP1 they gave you a list of bones that you needed to know and a list of muscles you needed to know? This is the vessels. Now, instead of staring at a model or staring at a picture and saying, okay, this is this vessel, that's that vessel, that's that vessel. It's easier to learn the blood vessels if we draw a map of saying that this vessel turns into this vessel, which splits into this vessel, which turns into this vessel. So the reason I gave you the paper is because we're gonna draw and diagram all of those vessels out. I promise you that if you learn to draw these, if you can draw these from memory, you will be like so much better off for the exam than just sitting here and trying to memorize and stare at that model, okay? So let me erase all of this and we'll start with the drawing.